Hi guys, I'm not as well dressed or as poetic as the previous guy, but we'll see what I can do. All right, so I'm Jonathan, you can call me Johnny. I'm from Imperial College, and today I'm going to talk to you about lipids on Mars. So there are two very important questions that I want to try and answer during the course of this talk. Firstly, can we understand the processes of preservation of biomarkers in Martian environments? And two, how can we use that to tell us more about and inform us about future life detection missions on Mars? And to do that, I'm going to talk about biomarkers, appropriate terrestrial analogs, and how they work together to give us an idea of the preservation potential of these environments. So first thing we need to ask ourselves is, what are we looking for? Because there's no point spending $1 million, billions of dollars on a rover to send to Mars when we don't know what we want to find. So the answer to that is, of course, biomarkers, which Bradley has kindly told us, and the definition of which is here. But the question is, what makes a useful biomarker? How do we learn to choose what biomarker we want? So, for example, let's say that you want to find out more about dinosaurs. Okay, what biomarker do you use for dinosaurs? And the answer there is bones. And there are two reasons why you use bones as a biomarker of dinosaurs. The first one is that they're highly resistant to degradation. Dinosaurs died millions of years ago, and you can still find bones in pristine conditions today. The second thing is that bones are highly diagnostic of different types of dinosaurs. For example, if you see the plates of a stegosaurus, then you know that they're from a stegosaurus and not from a T-Rex, for example. So similarly, when we look at microbial mats on Mars, which is what we think would have been the most evolved life on Mars, we look at lipids. So you guys already know what lipids are because Bradley told you what they were, but essentially they are fatty acids. They have a variety of functions in the cell, such as triglycerides as energy stores, glycolipids as signalers, and perhaps most importantly, phospholipids as the components of cell membranes, which means that as long as your organism had a cell membrane, it will have lipids. So similar to bones, lipids, the hydrocarbon derivatives of lipids are highly resistant to degradation, and lipids can be diagnostic indicators of different biosynthetic pathways or of different domains of life. So now that we've figured out what we want to look for, we need to figure out where to look for them. And in order to do that, we need to identify what the most common rock types that were deposited in various major periods of Martian history. And we look at that through the mineralogical eras, the Philosian, the Theician, and the Siderican. And we can further constrain where we look for these rocks by figuring out where there is evidence for liquid water. Now, the question is, how do we understand the processes that are occurring in the environments millions and millions of miles away? And we do this by using terrestrial analogs, which are locations on Earth that are assumed to mimic some, if not all, of the environmental conditions of Mars, such as geological, biological, chemical. And the reason why we do this is, well, we can't get to Martian rocks just yet. There's no sample return missions, and with Trump in charge, there may never be. So instead, we look closer to Earth, we try and understand processes occurring on Earth, and then extrapolate that to Mars. And so, for example, the Philosian, which comprises mostly of iron-rich clays that are derived from the weathering of mafic basalts, we use glacial soils or volcaniclastic sediments as simulants for this environment. Similarly, when we look at the Theician, which is mostly jarosites, which is an iron sulfate salt, because the Theician had a lot of sulfur outgassing, we look at acid mine drainage or sulfur streams. So, for example, this is one of the classical terrestrial analogs, Rio Tinto in Spain. And finally, in the Siderican, where there is mostly ferrihydrite and other anhydrous ferric oxides, we look at iron bogs and fens. Now, these are still a lot of variety of environments. So today, I'm going to focus on my work in the Theician environment and this particular terrestrial analog. So let's take a closer look at that. So this is a picture of my study area. There's a guy here for size, for scale. Geologists very careful about scales. Let's take a closer look at what a sulfur stream is. So this is a sulfur stream. Sulfur streams are caused by the dissolution of sulfides. Usually, it is pyrite into water. And what happens is that this eventually deposits jarosites, which are an iron sulfate salt. And this is very similar to environments formed on Mars, such as in Meridiani Planum. Now, as you can see very clearly, these environments are capable of supporting extremophilic organisms, ecosystems of these extremophilic organisms. And these are very useful to us because if these environments can support extremophilic organisms on Earth, then these same environments may have been able to support similar organisms on Mars. And if we can find out what these organisms look like and how they're preserved in Earth rocks, then we can figure out what they would look like if they had been on Mars and are preserved in Mars rocks. So once again, this is where my study area is. Of course, the best analogs for Mars are found in the south coast of England. Like most other sulfur streams, this sulfur stream is derived from pyrite in the Wealden beds in the surrounding geology. And like Meridianum Planum, the primary mineralogy is jarosite. But there's also goethite, which is an iron oxide derived from the transformation of this jarosite under humid conditions. Also present, of course, is the ever-present clays. Now, the organic inputs into this environment include acidophilic algae, microbial mats, which are primarily purple sulfur bacteria, as well as lignitic wood. And what we're trying to investigate is how the lipids from these organic inputs are preserved in rocks in these sulfur-rich environments. So this is how we do it. Firstly, we collect a bunch of samples. We took 9 different cores in order to sample the lateral and vertical variations in the stream. We then separate them by mineralogy, send them for XRD analysis, powder them so that we can prepare them for Bly dye extraction, which is a type of liquid-liquid extracting technique that will remove the lipids from these samples. We then derivatize them and put them on a gas chromatography mass spectrometer, aka the GCMs. Now, before I show you this GCMS data, let's take a look at what a core profile would look like in this sulfur stream. So at the very top, you have your microbial mat. Beneath that, you have a goethite layer that slowly transitions into jarosite. And at the very bottom, you have clays. So this is the GCMS data, where 3M represents a microbial mat sample, 3R represents a goethite sample, and 3C represents a clay sample. Now, not many of you may know how to interpret GCMS data. So what you're showing here is the GC data. Each of these peaks represents a different organic molecule, in this case, a different lipid species. And you can identify that lipid species by looking at the mass spectrometer part of the data, the MS part of the data. The height of those peaks represents the relative abundance of that particular lipid species to all the other peaks. And we usually use an internal standard, in this case the IS, to help give us a quantification of how much lipid there is in this sample. So what do we want to find out from this GCMS data? Well, obviously, we want to figure out what lipids are being preserved. Secondly, we want to figure out if there are any patterns in lipid preservation that would be able to distinguish them from abiotic sources. We then want to figure out what these biomarkers can tell us about the organisms living in these environments. And lastly, we want to figure out if there's a preservation bias. Are some lipids better preserved than others, and what are the factors that affect them? So before I go into the raw data, etc., let's remember what the different types of lipids are. So these are saturated lipids. They don't have any functional groups other than the carboxyl group at the end. And the other fatty acids, they are named depending on what functional group they are. For example, a double bond, an extra methyl group, or a hydroxyl group. So this is the GCMS data, except with colors this time. You have abundance on the y-axis, the different lipid species on the x-axis, and each of those colored boxes represents a different group or different family of lipids. Now, the one thing that should stick out to you is that there seems to be a much larger abundance and diversity of lipids in the goethite layer as compared to the clay layer. Now, for geologists, that seems really weird because clays are generally more associated with the preservation of organic molecules than, for example, an iron oxide. As you can see, the clays only possess C16 and C18 saturated fatty acids. So I'll talk about that later, but another thing I would like to point out to you is what we call an even over odd predominance in the carbon chain lengths of the saturated fatty acids. So the C14, C16, and C18 carbons are much higher in abundance than C15 and C17 carbons. And this makes sense because this is a configuration that's very preferred by terrestrial organisms. But what this means is that this is a biogenic pattern, right? You can't form this kind of pattern abiotically. So this is a pattern that will be good for distinguishing between biotic and abiotic signals. So next, let's take a look at what these biomarkers can tell us about the organisms that lived in these environments. So we can split them into two. There are general and specific biological indicators. A general biological indicator is something that is indicative of life in general. And usually these are the most well-preserved, and they should be ubiquitous in all the samples. And in this case, it would be C16 and C18 saturated fatty acids, because as you saw, they were found in all the samples, including clays. And this makes sense because they're the most common saturated fatty acids in all of organisms. The only thing is that these don't provide a lot of taxonomic data, so we look to specific biological markers to tell us that. And we can use these to figure out whether or not something was an anaerobe, an aerobe, a bacteria, or archaea. Now, some of these are not super useful. For example, we don't think that life on Mars would have developed aerobic metabolism or plants, so we can cross those out. But the rest of them are still very relevant in case we find any organic signals on Mars. Unfortunately, one thing you need to notice is that most of these are functionalized lipids. And what usually happens is that functionalized lipids aren't very well preserved. So this is the relative degree of preservation on the y-axis over the lipid diversity on the x-axis. And you can see that, so these are the functionalized lipids. And you can see that the relative degree of preservation is much lower for these lipids as compared to the saturated fatty acids. So although functionalized lipids are very good at telling us taxonomic information about the organisms that live in these environments, they may not be very well preserved on Martian environments, right? So this is a preservation bias that we need to take into account of when we look at Mars. So what other things could affect this preservation bias? Well, there appears to be a mineralogical bias, as we saw, because goethite possessed more fatty acids than did the clays. And that's really unusual. So there could be a number of reasons for this, right? The goethite contains more abundance and a higher diversity of lipids. But the implication of this is that organic material could be sequestered in what we call a rusty sink. These, the organic molecules could be attached to these ions, and they could be buried. And this mimics a terrestrial phenomenon where up to 21% of organic carbon in terrestrial sediments is bound to iron phases. And so the implication of this, and this is the one thing that you should take away from this talk, is that goethite and other iron oxides could be of astrobiological importance on Mars. So one more thing before I wrap up the talk. As you can obviously see, these lipids were found in an extant community. They were being produced in real time. We need to find out what these lipids look like 3 to 4 billion years later, right? Because Mars life likely existed only 3 to 4 billion years ago. So the primary process that we need to look out for is what we call deepcarboxylation. And this is a very simplified explanation of what it is, but essentially what happens is that your saturated fatty acids become alkanes by losing a CO2 group. And if you remember the even over odd predominance pattern that I talked about in the fatty acids, well, because you're losing a single carbon atom, what happens is that this predominance becomes an odd over even predominance in alkanes. And remember the general biological markers? Well, they become from C16 and C18 fatty acids, C15 and C17 alkanes. So let's go over the conclusions. Let's return to the two questions that we asked ourselves at the very beginning of this lecture. What are the processes of preservation of Mars, and can we understand them? The answer is, yeah, we can, because we can use lipids as biomarkers. We can select appropriate terrestrial analogs, in this case, sulfur streams for Theakian Mars. We know that certain species of lipids can be used as diagnostic indicators for different metabolisms or different domains of life, but that these are affected by the preservation of these species, depending on what species they are and what mineralogy is present in the analog environment. So how do we use this to figure out more about future missions to Mars? Well, we figured out that goethites and other iron oxides are possibly of astrobiological importance, and we've identified several potential biogenic signatures, such as C16 and C18 saturated fatty acids, as well as EOP or OEP patterns in fatty acids and alkanes, respectively. And thank you. That's the end.
Hi. For a given organism or organism group, up to how many different lipid signatures can result? Is it strictly 1-to-1, or could it be many to one?
It could be many to one. Mostly because basically many organisms basically produce a large amount of lipids, and the lipids I've shown you are only a small fraction of that number. There are many other different types of lipid biosignatures that could be present depending on the type of organism. And it's usually quite difficult to pinpoint a specific lipid to a specific organism, which is why sometimes you just have lipids that correspond to entire domains of life. Like, for example, a spin lipid could say, this is formed by bacteria, but we have no idea what bacteria it is.
In your preservation slide, your palmitic acid seemed to be a lot less preserved than the stearic acid,
yes,
which is just interesting,
given
that they're not very that they're very structurally similar, and also
Yeah.
given that those are the two that show up the most.
That is quite an unusual pattern that I observed. I'm still not very sure why that was the case, because C60 and C18 obviously, palmitic and stearic acids are obviously the highest, most abundant of the saturated fatty acids, all fatty acids in general. So it was quite unusual that we saw this particular pattern, and we're still not sure what caused it. If you have any ideas, like, I would love to hear them.
Yeah, I mean, just structurally, I can't think of anything.
Yeah, because structurally they are pretty much the same. All
Yeah.
that's happening is that you have an additional methyl group at the end, right? Sorry, additional 2 methyl groups at the end. So by right, there shouldn't be any difference in terms of preservation, but we do see that here.
Okay, cool.
So in a few months, MSL is going to arrive at Hematite Ridge in Gale Crater. If we see goethite as part of that ridge, how do you think they should sample it?
How do I think they should sample it? Well, I hope they don't blow it up. Yeah, well, yeah, there was another thing about biomarkers that that's a key point of biomarkers that I didn't explain, and that is that they should be easily analyzed in their geochemical environment. And one of the problems with the way that we analyze biomarkers on Mars is that we tend to blow them up because we use thermal pyrolysis. And that's actually quite a large problem on Mars. It's the reason why we can't sample much of Mars's surface because of the presence of perchlorates or other oxidants on the surface. Because what happens is when you have a strong oxidant and you have hydrocarbons and you put them together and you burn them, combustion occurs and you lose all your signals. So that's a very big problem. Obviously, I would like for there to be wet chemistry to do it, but that may or may not be possible because bring solvents over to Mars, etc., etc.
All right. Well, thank you very much.